To understand the biological engineering of the terrestrial plant, one must start with the scale of the modern results. A giant redwood, Sequoia, was much simpler. The ancestors of all plant life were aquatic, microscopic green algae. There is a 500 million year gap in structural complexity between a single-celled sphere of algae and a towering pine tree. Early classification systems, such as the one used by Linnaeus, categorized plants by gross superficial traits. They grouped species by their habit, color, and the number or shape of their leaves, or even the structure of the androecium. Modern phylogenetic taxonomy looks past these surface characteristics. It reorganizes the plant kingdom based on ancestral relationships and the specific evolutionary solutions organisms develop to survive. We will track the three major structural upgrades that allowed plants to colonize dry land: the development of anatomical differentiation, the invention of vascular tissue, and the evolution of the seed. This structural view of the phylogenetic tree records the incremental physiological solutions necessary for life to colonize the terrestrial environment. Algae represent the structural baseline of the plant kingdom. They exist almost exclusively in marine and freshwater habitats, where the environment provides both constant hydration and buoyancy. These organisms operate on a thallus body plan. Because they are submerged in their required resources and supported by the water, they do not need true roots, stems, or leaves to find water or fight gravity. This baseline thallus can take various forms, ranging from microscopic colonies like volvox to the filamentous strands of Eulothrix. Because their physical architecture is relatively simple, taxonomists classify algae by cellular biochemistry rather than gross morphology. The three main classes, Chlorophyceae, Phaeophyceae, and Rhodophyceae, are distinguished by their internal properties. They are identified by specific pigments, like fucoxanthin in brown algae, and unique carbohydrate storage methods, like Floridean starch in red algae. Their reproductive systems are also tied to water. Both asexual and sexual reproduction rely on flagellated zoospores. These motile cells require a liquid medium to swim, find a mate, and fuse. Algae are physiologically engineered for a liquid world. Without structural support or environmental water for fertilization, they are unequipped to survive exposure to dry land. Moving on to land introduced two immediate challenges: extracting nutrients from a solid substrate and surviving exposure to the air. The bryophytes, the mosses and liverworts, developed the first terrestrial upgrade. To survive on soil, they evolved rhizoids, unicellular or multicellular root-like structures used for attachment to a substrate. This graphic highlights the core division in bryophytes. The main plant body is the haploid gametophyte. It generates a smaller sporophyte that remains attached and derives nourishment from it. They developed multicellular sex organs, the antheridium and archegonium, but lack vascular tissue to transport water. This creates a critical reproductive tether. The male antherozoids possess flagella. To reach the egg within the archegonium, the flagellated male antherozoids must swim through a physical film of environmental water. These plants can live on soil, but are dependent on water for sexual reproduction. This makes bryophytes the amphibians of the plant kingdom, restricted to damp and shaded microclimates where standing water is available. Significant vertical growth appeared with the pteridophytes, a group including ferns and horsetails. This solved gravity. Pteridophytes evolved vascular tissues, xylem to pull water from true roots, and phloem to distribute nutrients. This triggered a life cycle inversion. Now the main plant body is the diploid sporophyte, relegating the gametophyte to a tiny independent stage. However, these plants aren't fully decoupled from water. The prothallus still produces male gametes that must swim through environmental water to reach the archegonium. Because fertilization still requires a liquid medium, the geographical spread of pteridophytes remains restricted to narrow, damp regions. While vascular tissue allowed plants to conquer the downward pull of gravity, the archaic reliance on swimming sperm prevented these organisms from achieving total terrestrial dominance. Colonizing arid environments required plants to sever their reproductive reliance on standing water. The gymnosperms, which include the conifers, achieved this through deep adaptations for temperature and humidity extremes. This diagram illustrates their reproductive solution, heterospory. Instead of uniform spores, gymnosperms produce distinct microspores and megaspores housed within specialized cones. The male gametophyte is highly reduced, shrinking into a self-contained, durable transport vessel called a pollen grain. Gymnosperms do not require a film of water. Instead, millions of pollen grains are released and carried by air currents to reach the ovules. When a pollen grain reaches the ovule, it germinates. A pollen tube grows through the tissue to deliver the male gametes directly to the mouth of the archegonia. Following fertilization, the ovule becomes a naked seed. This structure protects the embryo and provides a self-contained food source, allowing the plant to survive in a dormant state until environmental conditions are favorable. By utilizing the wind for transport and the seed for protection, gymnosperms permanently broke their reproductive dependence on liquid water. Reviewing Kingdom Plantae through these physical upgrades reveals the specific order of physiological challenges these organisms solved. This data shows the milestones, moving from a buoyant thallus to vascular roots, shifting the dominant life stage, and replacing swimming spores with wind-dispersed pollen. This leads to angiosperms, which evolved flowers and fruits to protect seeds and diversify reproduction. This systems-level view explains why modern taxonomy focuses on internal features, like anatomy and embryology, rather than the superficial traits used in the past. Every terrestrial plant is a living record of these engineering solutions, documenting how life systematically overcame the challenges of the dry land.